1. Gene-hacked mice produce their own Ozempic-like drug, the future of diabetes treatment. 2. Gene editing for weight loss, DIY drug delivery breakthrough. 3. CRISPR makes mice produce own Ozempic drug gene therapy for obesity and diabetes. 4. Revolutionary gene editing for weight loss, no more daily injections. 1. Gene-hacked mice produce their own Ozempic-like drug, the future of diabetes treatment. **** Imagine a future where your body manufactures its own medication, eliminating the need for daily pills or injections. ***That's the tantalizing possibility emerging from groundbreaking new research. And it's far more complex than it initially appears. Scientists are now successfully gene-hacking animals, specifically mice, to produce drugs similar to Ozempic directly within their own systems. This isn't just about weight loss, it's a potential paradigm shift in how we treat a wide range of conditions. A recent study, published in the journal Communications Medicine details how researchers at Japan's University of Osaka achieved this feat. They utilized a precise gene editing technique, based on CRISPR technology, to modify the livers of mice. The modification involved inserting a gene that instructs the liver cells to create Exenatide, a drug that belongs to the same class as Ozempic and Mounjaro, and was one of the earlier medications used to manage type 2 diabetes. Essentially, they reprogrammed the mice's own biology to become miniature pharmaceutical factories. The subjects of this study weren't just any mice. They were obese and pre-diabetic, mirroring conditions increasingly prevalent in human populations. After the gene editing, researchers observed a significant difference between the modified mice and a control group. The gene-edited mice demonstrably ate less food and experienced less weight gain over a period of 28 weeks, with the produced Exenatide consistently detectable in their bloodstream. This suggests the gene editing was successful in establishing long-term drug production within the animals. This isn't an isolated incident. Researchers have been exploring the idea of biopharmaceutical animals for years. Back in 2017, a team at the University of Chicago successfully gene-edited skin cells, both human and mouse, to produce GLP-1, the type of hormone that Ozempic mimics. These modified cells were then grafted onto animals, leading to increased insulin secretion and weight reversal. More recently, students at the University of Ottawa even managed to genetically engineer a plant relative of tobacco to produce a similar compound. But here's where it gets controversial. Are we comfortable with the idea of growing our medicine in living organisms? And what are the ethical implications? Collectively, these studies represent a compelling proof of concept demonstrating the feasibility of personalized, genetically driven treatments. Instead of relying on external pharmaceutical production, the body could potentially manufacture the medications it needs, offering a radical departure from the current healthcare model. As Keiichiro Suzuki, the senior author of the Osaka study, explained, the goal is to develop one-time genetic treatments applicable to conditions not necessarily tied to specific genetic defects. However, it's crucial to approach this research with a degree of caution. As Gizmodo points out, not all GLP-1 agonists are created equal. The original formulation of Exenatide, when taken orally, wasn't as long-lasting as newer drugs like semaglutide, Ozempic. While an extended-release version was developed, both original formulations were eventually discontinued due to concerns about pancreatitis, inflammation of the pancreas. And this is the part most people miss. Ozempic itself has also been linked to pancreatitis, although research on 2. Gene editing for weight loss, DIY drug delivery breakthrough. Revolutionizing weight loss, gene editing unlocks the potential for a DIY weight loss drug delivery system. Imagine a world where you no longer need to inject weight loss drugs every week, but instead, your body can produce its own. Sounds like science fiction, right? Well, it's not. A groundbreaking study from the University of Osaka has made this a reality by harnessing the power of gene editing to engineer mouse liver cells to secrete Exenatide, a drug used to treat obesity and prediabetes. The problem with traditional weight loss treatments. Obesity and type 2 diabetes are two of the most pressing global health problems, driven by complex combinations of diet, environment, and genetics. Traditional gene therapy has limitations when it comes to treating these conditions, as it works best when a single faulty gene is to blame. However, obesity and type 2 diabetes are complex conditions that don't have a clear genetic target, making it challenging to develop effective treatments. The promise of genome editing. Genome editing, particularly tools like CRISPR, has revolutionized the field of genetics by enabling precise modifications to the DNA sequence. While it has been successful in treating rare diseases caused by single mutations, its application to complex conditions like obesity has been limited. However, researchers are now exploring the potential of genome editing to insert new genes that can produce therapeutic proteins, such as Exenatide, which can help with weight loss and blood sugar control. How genome editing enabled sustained weight loss drug delivery. In this study, researchers used a genome editing method called homology-independent targeted insertion (HITI) to insert the modified Exenatide encoding gene into mouse liver cells. The goal was to create a one-time treatment that would turn the liver into a long-lasting source of the drug, eliminating the need for injections. To achieve this, they packaged the Cas9 enzyme and donor DNA into lipid nanoparticles, similar to those used in some mRNA vaccines, and delivered them via a single intravenous injection. The results. The study showed that the genome-edited mice produced high levels of Exenatide that could be detected in their blood for several months after the introduction of the gene. The mice ate less, gained less weight, and had better blood sugar control, with improved insulin sensitivity and reduced HbA1c levels. The treatment also didn't cause liver damage or interfere with normal GLP-1 signaling. What this means for the future of weight loss treatment. This study has significant implications for the treatment of obesity and other conditions managed with biologic drugs. By harnessing the power of genome editing, researchers may be able to develop a one-time treatment that can turn the liver into a steady source of therapeutic proteins, reducing or replacing the need for repeated injections. While there are still limitations to this approach, including the low editing success rate in the liver and the potential for immune system reactions, this study offers a promising new direction for the treatment of complex diseases. The next steps. The researchers plan to refine the editing process and test the treatment in other animal models, including those that more closely mimic human obesity. If these hurdles can be cleared, genome editing could become a practical tool for managing chronic diseases that don't have a clear genetic cause. References. Hirose J., Aizawa E., Yamamoto S., UIS, Suzuki K. Targeted in vivo gene integration of a secretion-enabled GLP-1 receptor agonist reverses diet-induced non-genetic obesity and prediabetes. ComsMed, 2025, 5/1-269. doi 10.1038 S4385/025-00959-8. 3. CRISPR makes mice produce own Ozempic drug gene therapy for obesity and diabetes. Imagine a world where your body could produce its own life-changing medication, no injections, no pills, just a one-time tweak to your DNA. Sounds like science fiction. A groundbreaking study from Japan suggests it might be closer than we think. Researchers have genetically engineered mice to manufacture their own version of Ozempic, a popular diabetes and weight loss drug, raising both excitement and questions about the future of medicine. But here's where it gets controversial. Could this approach one day eliminate the need for pharmaceuticals altogether? And if so, what does that mean for our bodies, our health systems, and our wallets? Let's dive into the details and the debate. The study, published in Communications Medicine focused on Exenatide, a first-generation GLP-1 receptor agonist. Scientists use CRISPR gene editing technology to modify the livers of mice, enabling them to produce Exenatide internally. The results. For 28 weeks, the treated mice maintained therapeutic levels of the drug in their blood, lost more weight, ate less, and showed improved insulin sensitivity compared to their unmodified counterparts. Importantly, no harmful side effects were observed, including no disruption to their natural GLP-1 hormone production. This proof-of-concept experiment hints at a future where gene therapy could replace traditional medications for chronic conditions like obesity and diabetes. But wait, GLP-1 drugs like Ozempic and Wegovy, which contain semaglutide, are already revolutionizing treatment. Why fix what's working? The answer lies in the limitations of current therapies. Semaglutide, for instance, requires weekly injections, which some patients struggle to maintain. Older versions of GLP-1 drugs, like Exenatide, demanded daily doses. Compliance is a major hurdle, compounded by high costs and gastrointestinal side effects. Pharmaceutical companies are already racing to develop longer-acting formulations, but the Japanese team took a different route. Instead of extending drug half-lives, they reprogrammed the body to become its own pharmacy. This isn't just about convenience, it's about sustainability, explains Keiichiro Suzuki, the study's lead researcher. The team's approach involves a single treatment that could, in theory, provide lifelong benefits. However, the idea of editing human cells to produce drugs raises ethical and safety concerns. For example, what if the body overproduces the drug, leading to unintended consequences? Or what if the therapy's long-term effects remain unknown? These questions are far from hypothetical. Gene therapy is still in its infancy for non-genetic diseases, and the potential for lifelong side effects remains uncharted territory. Fractal Health, a biotech startup, is already testing similar ideas. Their experimental gene therapy targets pancreatic cells instead of the liver, and early trials in mice showed weight loss exceeding that of semaglutide. The company recently filed for approval to begin human trials in Europe, a move that underscores the growing interest in this field. Yet, skeptics argue that gene therapy's risks may outweigh its benefits for conditions that can be managed with existing treatments. After all, why gamble with DNA when a weekly injection, however inconvenient, is already effective? So, where does this leave us? For now, GLP-1 drugs like semaglutide will remain the gold standard. But the Japanese study, and others like it, open a door to a radical new paradigm. Instead of taking medication, our bodies could make it. The question is no longer if this technology will work, but when we'll be ready to embrace it. And here's a thought to chew on. If you could choose between a single, permanent treatment, or a lifetime of injections, which would you pick? Share your take in the comments, because this is the part most people miss. The future of medicine isn't just about what we can do, but what we are willing to risk to do it. 4. Revolutionary gene editing for weight loss. No more daily injections. What if your body could become its own pharmacy, producing life-changing medications without daily injections or endless pills? That's the revolutionary promise of a groundbreaking study from Japanese researchers, who've unlocked a potential game-changer for weight management and chronic disease treatment. But here's where it gets controversial. This isn't about fixing a single faulty gene. Instead, it's about teaching the body to manufacture its own therapeutic proteins, effectively rewriting the playbook for how we approach obesity and diabetes. And this is the part most people miss. The innovation doesn't rely on correcting genetic mutations at all. Instead, it introduces an entirely new gene to create a built-in solution. The research, set to appear in Communications Medicine comes from a team at the University of Osaka. They are tackling the tricky challenge of non-genetic, complex diseases like obesity and type 2 diabetes, conditions that don't stem from one clear genetic cause, but still devastate millions. Traditional genome editing, which typically targets specific mutations, often falls short here. So, the team took a bold leap. Instead of fixing broken genes, they added a brand new one. This gene, when inserted into the liver of lab mice, instructed the body to produce Exenatide, a powerful weight loss medication that mimics natural hormones to regulate appetite and blood sugar. Think of it as turning the liver into a tiny drug factory. Once the gene was introduced, the mice began churning out Exenatide for months, eliminating the need for repeated injections. The results: the treated mice ate less, gained significantly less weight, and showed dramatic improvements in glucose metabolism and insulin sensitivity, all without harmful side effects. Keiichiro Suzuki, the study's senior author, called the findings very exciting, emphasizing how this approach could transform treatment for diseases with no clear genetic roots. But let's pause for a moment. What if this technology shifts the medical landscape so drastically that we begin to question the ethics of altering biology for non-genetic conditions? After all, obesity and diabetes are often linked to lifestyle factors, not DNA. Does this breakthrough empower patients, or does it risk oversimplifying complex health issues? We'd love to hear your thoughts in the comments. Do you see this as a miracle solution, or a Pandora's box waiting to open? The implications stretch far beyond weight loss. By creating a sustainable, in-body drug production system, this method could reduce the burden of chronic illness, improve medication adherence, and even lower healthcare costs. For example, instead of injecting GLP-1 receptor agonists weekly, patients might receive a one-time treatment and let their bodies do the rest. The study's mice maintained therapeutic Exenatide levels for months, proving the concept's staying power. Suzuki's team envisions applying this technique to other conditions that elude traditional genome editing, like chronic inflammation or metabolic disorders. Imagine a future where a single treatment could manage multiple symptoms, from obesity to prediabetes, by leveraging the body's own machinery. While the research is still in early stages, the potential is staggering. Of course, the road ahead isn't without hurdles. Scaling this technology for human trials will require rigorous safety testing, and ethical debates are sure to follow. But for now, the study offers a tantalizing glimpse into a world where the body's own cells become allies in the fight against disease. As the scientific community digests these findings, one thing is clear: we are standing at the edge of a new era in medicine, one where the line between treatment and self-sufficiency blurs, and the future of health care might just be written in our genes.